This is Mix 2 in the review series. In this presentation, we're going to look closely at the biomolecule nucleic acid. We're also going to look at the process of replication and protein synthesis. First, all life begins with the nucleic acid monomer, the nucleotide. This beautiful nucleotide is made up of a sugar, a phosphate, and a nitrogenous base. One of the easy ways to remember that the sticky sugar is in the center is that sugar can be sticky. And so the phosphate sticks to the sugar on one side and the nitrogenous base sticks to the sugar on the other side. Let's look at the structure a little bit more closely. I told you that this is a nucleic acid, but can you verify this by checking the elements? Do you see the CHON, a carbohydrate, a hydrogen, an oxygen, and a nitrogen? One of the trickiest things to telling the difference between nucleic acids and the protein is that one P in the phosphate group. So make sure you can locate that P, and here it is. So this is a nucleic acid. There are two main types of nucleic acid, DNA and RNA. They both have similar structures, but there are some very easily noticeable differences. DNA is the code for all life. It contains the code for making proteins, which is pretty much everything that makes up who you are. DNA cannot leave the nucleus, and how much DNA an organism has can indicate how complex the organism is. For example, bacteria has very little. DNA has the sugar base deoxyribose. It is a double-stranded helix, or coil, spring shape. DNA has the 4 nitrogenous base pairs, which are adenine, thymine, guanine, and cytosine. Notice how the backbone or the sides of this ladder pictured here consist of the phosphates. Here are the phosphate, here's a P. And it also consists of the deoxyribose, or the sugar. Here's your sugar. The rungs of the ladder, pictured here, are made up of your nitrogenous bases. Here's your adenine, your thymine, guanine, and cytosine. The bases are also going to pair together very specifically. A with T, as pictured here, G with C, as pictured here. And there's not going to be an exception to this. An easy way to remember this is apples in the tree and cars in the garage. At the center of the DNA is this bond right here. This is your hydrogen bond. It's a fairly weak bond that holds your DNA together. The sequences of nucleotides, A's, T's, C's, and G's, is going to form a unique genetic code. This unique code is unique to every organism. The closer the relationship is between two organisms, the more similar their DNA nucleotide sequences will be. Scientists can use these nucleotide sequences to determine evolutionary relationships among organisms. They can also determine whether two people are related and identify bodies at crime scenes. DNA spends most of its time uncoiled in a stringy form known as chromatin. However, during part of the cell replication cycle, mitosis, the DNA will coil around proteins called histones for easy packaging and dividing. This tightly wound structure is called a chromosome. You can kind of think of it like string wrapped around a spool. We'll talk more about that during mitosis. RNA also has a phosphate backbone, but there is only one phosphate backbone, not two like with DNA. Because of this, RNA does not have base pairs. It has nitrogenous singles waiting to find a match. This will be very important when we get into protein synthesis. RNA still have the nitrogenous bases guanine, cytosine, and adenine. However, thymine is replaced with uracil. Adenine will now pair with uracil. Guanine still pairs with cytosine. You can think of it like apples under the tree, and cars are still going to be in the garage. There are 3 different types of RNA. Ribosomal RNA, this is what makes up the ribosome structure in your cells. Messenger RNA, or mRNA, this is a special message that is created from your DNA, and it can exit the nucleus and search out ribosomes. Transfer RNA or tRNA. These special RNA carry amino acids on one end and an anticodon on the other. They are designed to find their matching codon and transfer their amino acids into a long chain that will eventually make a protein. So let's sum up the differences between DNA and RNA. DNA has the sugar deoxyribose. RNA has the sugar ribose. DNA has thymine, RNA has uracil. DNA has two strands coiled up into a helix, and RNA is usually single-stranded. DNA forms a double helix, and RNA does not. See if you can remember all of the differences between DNA and RNA without looking at your notes. Do you remember how the bases pair up? And which can leave the nucleus, DNA or RNA? Have you ever wondered why when you cut yourself, your skin heals back and it looks exactly the same as before you cut yourself? Well, in order for your body to heal, you have to make new cells. But those cells need the same genetic information that all of your other cells have. Otherwise, they won't be identical. All your new cells have to have an exact copy. Not a sort of kind of copy, but an exact copy. In fact, every cell in your body has to have the exact same copy of DNA, no matter what that cell's function is. So your skin cells have to have the same DNA that your heart cells have, even though they look very different and have a totally different function. Remember, your DNA does not leave the nucleus. So how did the cells get a new copy of this DNA? Well, your original cell's nucleus must make it before dividing, so it can give it to the new cell. And this whole process is called DNA synthesis, or DNA replication. DNA replication happens in a four-step process. The first thing that's going to happen is your DNA strand is going to uncoil. Once it's uncoiled, the enzyme DNA helicase will unzip your DNA strand. Basically, it breaks apart the hydrogen bond, much like unzipping a pair of pants. It's going to break the hydrogen bond between the nitrogenous bases so that all your A's, T's, C's, and G's detach and are exposed. Next, the free-floating nucleotides in your nucleus are going to come over and find their complementary base pair. Remember, apples in the tree, A pairs with T, cars in the garage, your G's are going to pair with C. As these pairs bond, a new backbone of the sugar and the phosphate is also going to form. So if you'll take a look at this ongoing DNA replication image, you'll see a couple of things that were mentioned in the 4 steps. First thing I want to point out is the original DNA strand, or this portion of the original DNA strand. The portion where the DNA splits is called the replication fork. It's right here, denoted by this key area, replication fork. The next thing I want to show you is your original backbone. Now notice your original backbone is here in the teal. This is one side of the original backbone. And over here, this is the other side of the original backbone. So if you could imagine for just a moment that both of these were put back together, that would have been the original DNA strand. Now if you look here in the center, you can see the newly forming DNA backbone here, and you have another newly forming DNA backbone here. The reason why this is so significant is because we would say that the end result of DNA replication is 2 new DNA strands, but each new DNA strand has half of the original parent DNA, but also half new complementary DNA. So in this way, you can keep the cells consistent. Each new cell has half of the original DNA from the cell before, but then the other half of the DNA is made from those complementary base pairs. The last thing that I want to point out before we move on is those matching base pairs. So remember we said apples in the tree, here's an A and a T. We say cars in the garage, so here's a G and a C. All right, so I have a C right here. What's going to pair with the C? I hope you said G. There we go. All right, so let's check out the next one. I have a T right here. What would pair with the T? I hope you said A. All right. Let's do one more. Let's do this A over here. Let's do this A. All right, good. It would be a T. Proteins are essential to the makeup of any organism. In humans, almost everything that makes up who you are is a protein. Remember, proteins include structure proteins like your hair and your nails. They include your immune system, your antibodies, and all of your enzymes, which build up and break down all the chemical reactions in your body. Without proteins, life would not exist as we know it. The trick is, how do you make them? And what tells the body what proteins to make? If your cell is going to make these proteins constantly and make them correctly, there must be some sort of directions and a way to make sure that they come out right each time. There are two parts to the protein puzzle. Transcription comes first, and then translation. Transcription is the making of a small piece of directions, or like the blueprints to making a closet in a large house. Translation is the process of reading those blueprints correctly to make the perfect protein, or closet in this case. Let's first look at transcription. DNA is the master code and holds all the instructions that make up anything and everything in your body. But where is the DNA located? It's located in your nucleus. It's trapped there and it will not come out. So the question is, how does it make a protein that either becomes part of your cell's functions or a protein that leaves your cells to carry out other functions? Well, the answer is the proteins are actually made outside of your nucleus. They are made in part by little tiny organelles called ribosomes. So what's the problem? We have the instructions to make proteins in the nucleus, but they are actually made outside of the nucleus. So what's the solution? DNA needs some type of messenger to carry out the information. It needs to carry the information from inside the nucleus out into the cytoplasm of the cell. This creation of a special message is called transcription. Remember that there are three different types of RNA. There's mRNA, tRNA, and rRNA. mRNA is made during a process called transcription, and transcribe basically means to copy. So you can kind of think of it like a process like a copier is copying a piece of paper. There are four basic steps to transcription. The first step, the DNA strand is going to uncoil and unzip, just like it did in replication. Next, free RNA nucleotides, which will connect to the nitrogenous bases, adenine, guanine, cytosine, and uracil, will float up and attach to one side of the DNA strand. As they start attaching, they will form a new mRNA backbone, but the original DNA backbone will remain. You can see this here. Here in the center is the mRNA backbone. Remember how your nucleotides connect? Your sugar is sticky, so the phosphate sticks between the sugars. This is actually the wrong sugar. If we're creating an mRNA strand, what kind of sugar will we be using? I hope you said ribose. Remember, we're dealing with RNA, so we're going to have a ribose sugar. Each of these are going to be ribose sugars along here with a phosphate. And then your nitrogenous base is going to be stuck to that sugar as well. Remember, there is no thymine, so you will only have A, U, C, and G. For RNA, apples will go under the tree, but cars will still go in the garage. If we were to look at this individual side of the DNA here, we have a DNA strand here, and we have RNA starting to form over here. If we were to look at this, we can see how these will connect. This RNA A is connected to this T. Okay, so we have apples on the RNA. This is still a DNA strand, so this is still going to be a T, A to T. But over here, we have an A on the DNA side. Which one of these ribonucleotides will match up to the A? Remember, apples under the tree, so you can kind of see this U already coming to it. So A is going to match with U. This C is going to go to this G. This C is going to go to this G. This G is going to go to this C, and so on. Once the mRNA strand is complete, the hydrogen bonds that were holding it to the original DNA are going to break. So yes, an enzyme is going to be involved here, just like as when we did replication. The DNA strand is going to go back together as soon as that is done. Once it disconnects, the free mRNA strand is going to leave the nucleus through the tiny little pores and go into the cytoplasm. From there, it will attach to a ribosome. So again, you're going to have hydrogen bonds breaking along here, and then your mRNA strand will leave the nucleus and go out into the cytoplasm. The image that you see here is one of the most often images, or most common images, used on tests to show the process of transcription. And it's a very clear picture because it shows all of the unique differences between DNA and RNA. I want you to take a moment and look at the picture and see if you can identify the key differences between DNA and RNA. What are some ways that you can immediately look at this picture and see that what this process is and what it's making? Okay, so hopefully you pause the video for a second. All right, so if you look here, you can tell that this has 1 and 2 strands. So this is going to be DNA. You can also tell this is DNA because you have a T here. Okay, so that already tells you that what we're looking at is DNA. So the big question is, am I replicating or am I transcribing? How do I tell the difference? Well, if we're looking at the nitrogenous bases, down here, this has a U. So if you remember that mRNA or RNA in general will have a U and DNA will not, then you know that this is RNA. The other way you can tell is that this is single-stranded. It doesn't have a double strand, so this is definitely RNA. Now, if you're looking out here at the strand after it left, it's a single strand. It only has a U here, no T's, so this has to be RNA. Because of this, we can tell we are transcribing and not replicating. We are not creating a new strand of DNA. We're creating a single copy or single side of the DNA called mRNA. All right, so we know that the DNA is the boss of the cell. It has all the master instructions, the code for everything that you are. But what exactly is the role of mRNA? What is its purpose in the cell? Well, the mRNA carries a message. Remember, we're eventually going to be building proteins. So we can kind of compare the transcription process to other things that we want to build, like houses, for example. So if we were going to build a house, we would need blueprints. The mRNA is kind of like the blueprints for making a protein, because mRNA carries the instructions for how to build a protein. It will give the order for how the amino acids are going to connect, just like blueprints give the instructions to the construction team of how to build the house. Transcription is the first step to making a protein. Just like you can't build a house without the blueprints, you can't build a protein without the mRNA. The mRNA is the blueprint to making a specific protein, and that blueprint comes directly from the DNA. The next step to making a protein is translation. Translation is a word that describes all the events that happen from the time the mRNA leaves the nucleus to when the protein molecule is completed. It is a very busy process, and it takes all 3 types of RNA to complete. The first type of RNA is mRNA. Remember, mRNA is the blueprint to making the protein. It comes directly from the DNA, so it has to bring these instructions out to the cytoplasm. It kind of like delivers the blueprints to the job site. Next is the tRNA. tRNA is also called transfer RNA because it carries 1 amino acid on it. The tRNA has 1 amino acid at the top, but it also has an anticodon at the bottom. This anticodon is how the tRNA is going to bind with the mRNA. Remember, the anticodon is part of the tRNA, so it will have uracil instead of thymine. Let's look at the anticodon here at the bottom of this tRNA. We have UAC, and remember this anticodon is going to find a codon on the mRNA. The last piece of the translation puzzle is rRNA, also known as the ribosome. The ribosome is the structural site where the mRNA and tRNA meet to process the amino acid chains and eventually create a protein. All three of these RNA are required to make a protein, and each of them have a specific job in the process. All right, so let's break down the roles of each of the RNA. If we were to use our house building analogy, the mRNA would be what? Well, it's going to bring the instructions from the DNA to the ribosome, much like bringing a blueprint to the job site. You cannot make a house without a blueprint, and you can't make a protein without mRNA. But what else do you need to build a house? You need things like wood, nails, and eventually bricks or something, right? You need the supplies. So the tRNA is going to bring the supplies, just like construction worker would. It's also going to be called the transfer RNA because it transfers the amino acid onto the long polypeptide chain of amino acids. The final piece is the ribosome. The best way to describe the role of the ribosome is that of the building site. The builder gets the blueprints, the mRNA, then the supplies, the tRNA, and then they all get together and construct proteins at the building site, or the rRNA. So these are the parts. The next big question is the how. Remember that creating proteins is all about reading a code. Every 3 letters on an mRNA strand is called a codon. The word codon sounds a lot like code, doesn't it? Well, that's because a codon codes for something. It codes for a specific amino acid. In order to read the code, you need to use a codon chart. There are 2 basic styles of codon chart. There's the circular and the square, like the one here. They are both read in basically the same way. As you're reading, every 3 letters, each set tells you what amino acid you will be dropping off. That codon matches to the anticodon on the bottom of the tRNA. So let's look at an example. Let's start with codon 1 up here. We have GCU. I'm going to show you how in the next couple of clips how to read the 2 different codon charts. But I just want to simply show you on this one while I have both together. This side of the codon chart is the first side that you start on. A lot of times it's called first base. It's called that because this G is the first nitrogenous base that you see. So that tells you to start here along this line. Next, up here at the top, we have what's called second base. That's because we're going to look at the second base, which is C. Right? So find the C. And what this tells us is if we start with G, we're going to come all the way across. We're doing something along this row. The second base C tells us we're doing something along this row. So for right now, using the first 2 letters of the codon, we know we're in this box here. Now this particular codon chart does not have a third base box, but it actually tells you inside the boxes which codons are going to code for what. So if I were to erase this, all right, so if you look at the box that we narrowed down, it tells you which 3 letters or which codon are going to code for what amino acid. So we have GCU, we would look at GCU, and it codes for ALA or alanine. All right, let's look at another type of codon chart. This is the circular one. And let's say you have a codon, let's use A, G, C. That's my codon. And we're just going to use this just so I can show you how to use the circular one. All right, so in the circular one, first base is in the first ring. So this is first base. So my first base, number 1, is A. So I'm going to circle A. That means I'm going to be somewhere in this part of the pie. Right? Next base, second base is G. So second base ring is here. And my G is right here. So that tells me I'm narrowing the pi down to this area right here. Now I'm going to do my third base, which is C. Third bases are along here. My C is right here. So that tells me I'm going to code for serine. Now I want you to notice something. Here, I'll use a red pen again just to show you. Notice how I can either have U or C code for serine. So actually, if I had a codon of A, G, U as my codon on my mRNA, both of these would code for serine. So it is possible to have a codon code for multiple amino acids, and this is going to be really important when we start talking about mutations. So the codons on an mRNA strand need to be converted. They have to be read, and then they're going to be converted into amino acids in a fairly simple process. Remember, it requires not just the mRNA strand, but also the ribosome. And these right here are depicting an image of the tRNA. Remember, your tRNA has the amino acid on the end. So the process of converting these codons on the mRNA into a sequence of amino acids in order to make a protein is known as translation. So we've just completed the entire process of protein synthesis, which begins with transcription, the creation of the mRNA strand, and ends with translation, the reading of that mRNA strand to create the protein. I want to summarize protein synthesis and the rules of each of the RNA. This is what the entire ribosomal complex looks like as it's working. I want you to notice how the tRNA are connecting with the mRNA. Notice that the anticodon at the bottom of the tRNA is going to pair with the 3 bases on the mRNA. On the mRNA, those 3 bases are called the codon. An mRNA strand is always going to be read in sets of 3 nitrogenous bases at a time. So read 3, attach, read 3, attach, move on. As the tRNA attaches, it's going to drop off an amino acid. It stays in the complex until the next the next tRNA attaches. The new tRNA is going to drop off the next amino acid, and together these 2 amino acids are going to begin to form a polypeptide chain. You can see the beginning of a polypeptide chain in this image, in this little video. We actually have a total of 3 amino acids connecting, but again, keep in mind that the first tRNA isn't going to be released until the next tRNA comes along to attach the amino acid. So this whole process is going to continue until a stop codon is read. And when that is read, the polypeptide will form its final protein shape and detach and go about its tiny little protein life. So the big question is, how do you tell the difference between transcription and translation on a test question? We've already looked at how to tell the difference between replication and transcription on a previous clip. But what I want you to do when you get a test question like that is focus on the recognizable differences between DNA and RNA, just like we did before. First, DNA cannot leave the nucleus. So knowing that, we already can tell that this has to be DNA. Let's say that nothing on here was labeled. Let's say this DNA wasn't here. How do you know this is DNA? Well, one way I just told you, DNA cannot leave the nucleus. But also, look at the strand. How many strands do you have here? Second, DNA is double helix, okay? But the RNA is single helix, and you can actually see the RNA right here detaching. So this process right here is transcription. We're actually creating an mRNA strand, and it's leaving the pore and going out into the cytoplasm. All right, so if we were to look at the other part, I'm going to switch to a green pen here. If we were to look at the other part, we can actually see a huge complex in here. We can see the ribosome. We can see the little tRNAs. We can see the polypeptide chain. We can see the individual tRNA with the individual amino acids. So this whole entire complex, we're actually in the process of creating the protein itself. So this is translation right here, translation. I've also taken the time to summarize for you the basic steps of protein synthesis from the beginning with transcription all the way to the end of translation. So make sure you double check your worksheet and take the time to answer all the questions there. On the last slide here, I want you to practice everything that we've learned. So below is a strand of DNA. I want you to think for a moment as to what letters would connect with what. Pause the video here. On a scratch sheet of paper or on your worksheet, write down this code, and I want you to figure out what would transcribe, what would the mRNA line be. Okay, so now that you've taken the time to do that, during transcription, we're looking at what mRNA strand would be made from this DNA. So I'm just making sure you had time to pause the video before I do my big reveal. And here's my big reveal. All right, so again, make sure you remember that this is the DNA. And we know this because it has a T. We know this is the RNA because it has a U. If you made a mistake in the transcription process, usually the first thing that happens is you forget to use that U. Just remember, when I'm going from DNA to RNA, I'm going to use apples on top of the tree for DNA and cars in the garage, and apples under the tree for RNA and cars in the garage. All right, the next thing I want you to do is going to be a little bit tricky, but once you have your mRNA strand written down on your paper, I want you to pause the video and slide it back until you find one of those mRNA charts. Just pick the one that you would like to use, and I want you to translate this mRNA code into a protein or amino acid chain. All right, so what you will have on the bottom is underneath each of these three here, I want you to write the three-letter word or three letters that represent the amino acid. So again, you're going to translate this mRNA line using the codon chart. So go ahead and pause now and do that. Okay, I hope you paused and had time to do that. So we've used the mRNA strand to translate the codons into amino acids using the codon chart, making sure you had enough time to pause. And here's our big reveal. These are the amino acids, the 6 that you should have come up with. Thank you for taking the time to watch this video. Make sure that you fill out your sheet. If there's anything along the video that you missed, you simply pause and rewind, or ask a biology teacher in the room.